Доброго дня, шановні колеги. Ви
Hello, dear
підключаєтеся на
colleagues.
вебінар Центру
You are
громадського
now
здоров'я
joining the
спільно з
webinar from the
ICAP
Ukrainian Public Health
Зараз,
Center,
доки наші
which is
колеги
jointly
долучаються до
organized
цього вебінару,
by
ми
ICAP
почекаємо
and
і
estimated
виконуючи
colleagues.
веселунку будемо
As our colleagues
починати.
are joining,
Отож,
we are now
доєднуйтеся та
preparing
кличте своїх
all the
колег та
technical
знайомих,
parts.
які
So
планували
please
відвідати цей
get
вебінар.
connected. Please remind your colleagues who were willing to join this webinar, and please stay tuned.
Ще раз всіх вітаю. Я бачу, що до нас доєдналося вже
Hello
більш
once
ніж
again,
200
everyone.
глядачів.
I see we now
Всім
have more
доброго
than
дня.
200
дня.
viewers
Ви
available now,
долучилися
more than
до
200
вебінару Центру
participants. Hope
громадського
your day
здоров'я.
is peaceful.
Цей вебінар
This
є
is a webinar
продовженням
organized by the Ukrainian
циклу
Public Health
лекцій
Center. This is a
«Європейський досвід у
continuation of a
проведенні
series of
інфекційного контролю
lectures
та адміністрування
dedicated
антимікробних
to the
препаратів
European
перспективи
experience
для
of
України».
antimicrobial
Хотілося б нагадати,
resistance
що
control
перший
and
вебінар
IPC
вже триває з 23-го
experiences
року.
for Ukraine.
Запис
Let me
цього
remind you, we
вебінарів,
have
який
this
ми
series of
постійно
webinars since 2023,
записуємо та
and
викладаємо на
we have
YouTube-каналі Центру
videos
громадського здоров'я,
which are well available
виконується. Ви
at the
їх можете
website of
передивитися
the
на
Ukrainian Public
YouTube-каналі
Health
Центру
Center. You
громадського
can also
здоров'я.
use the
Цей
YouTube
вебінар і
channel of the
подальші
UPHC.
будуть викладені,
The given
записані,
webinar,
тому
as
ви
well as
завжди
all
маєте
the
змогу
rest
їх
of the
передивитися та
webinars in
ознайомитися.
series, they will be recorded and they will be also
Хотілося б окремо
published for
висловити
further
слова
use.
подяки нашим
I would like to
партнерам.
express my words of gratitude to our
Сполучених
partners,
Штатів Америки,
the
ICAP, Київського
the
університету
American
та
CDC Center,
організації ESCMID,
ICAP at the
лектори та
Columbian
досвідчені
University,
фахівці
ESCMID
якої
colleagues,
дозволяють та
because these are
допомагають
the
нам у
experienced
проведенні
lecturers
цього
and
циклу
users
вебінарів. На
that
цьому
are
вебінарі
often engaged into
передбачено
these webinars.
веб-переклад. Ви можете
During the
побачити
webinar, we have the
налаштування.
interpretation option
Вам
available.
треба буде
In order
включити
to use interpretation,
планетку і обрати
you need to
українську
go
мову,
to a
щоб
globe-shaped
слухати
button
цей
below
вебінар
and
українською
select
мовою. Знову ж таки,
appropriate
якщо є
channel
змога,
for your
можете
convenience.
передивитися його
And
і в
if you
оригіналі.
need, you can always watch
Лекція
it
буде
again
тривати
in
близько
English
години.
or Ukrainian.
І після чого,
The
в
estimated
залежності
time for
від
the lecture
того,
is
скільки у вас
around 1
залишиться
hour,
часу,
and
ви
then,
зможете
depending
поставити
on the
у
amount
чат
of time left,
запитання до
you will
спікера,
be able
і
to use the
наскільки
chat
дозволяє
box to
регламент,
ask
ми
some of your
спробуємо
questions
відповісти
to the
на
speaker.
якомога
And
більшу
then, if time
кількість
allows, we will
запитань.
try to give answers to as many
Тож,
questions as possible.
далі хотілося б проговорити, що до
Now,
кінця
let me
2025 року
briefly
заплановано
inform you that
ще
until
декілька
the end of
лекцій. Далі вони будуть
2025, we
більш
still have a couple
заплановані
of
для
lectures
колег
on our plan.
бактеріологічного
These are going to
профілю,
be
щоб не
mostly focused
пропустити
on
подальші
bacteriology profile colleagues.
вебінари, щоб не
In order
пропустити
not to
подальші
skip,
лекції.
not to
Слідкуйте за ресурсами
lose
YouTube
this
каналу
chance to
та
join our
особливо
webinars,
Facebook
please watch
каналом
our
Центру
web
громадського
resources
здоров'я, де всі
and the
такі
Facebook channel
анонси
of the
будуть
Ukrainian Public Health Center.
своєчасно
We're going to
опубліковані.
provide you with all
Отож, не
the
буду вас
announcements
затримуватися. Сьогоднішнє
in
дня вам
advance.
буде
So, without
передбачати
further ado,
методи визначення клінічно
I would
важливих
like
механізмів
to
протимікробної
invite you to the
резистентності в
further
gram
discussion
негативних
of our
бактерій.
today's
І
topic,
сьогоднішню
which is
тему
detection
нам
of clinically
проговорить дуже
important
досвідчений
resistance
фахівець,
mechanisms in
доктор
gram-negative bacteria.
Onur
And
Karatuna,
Dr.
який
Onur
працює в
Karatuna,
лабораторії
a very
розвитку
experienced
ЕОКАСТ
MD,
в
who will speak
Швеції.
about that. He
І
works for the EU
він
Cost Development
має
Laboratory,
дуже великий
the
фах
city
та
of
досвід у
Växjö,
методах
Sweden.
визначення
He is very well
мікробіологічної
experienced
чутливості
in
мікроорганізмів.
microbiological
Він
susceptibility testing
співпрацює
for microorganisms.
з Всесвітньою
He
організацією охорони
cooperates a lot
здоров'я
with
і
the
є дуже
WHO,
провідним та
and
досвідченим науковцем
Dr.
з точки зору
Karatuna is considered to
мікробіології.
be really
Отож,
the leading expert in the field of
докторе Каратуна,
microbiology in
слово
the
вам.
given scope. So, Dr. Karatuna, please, the floor is yours.
Inat, thank you very much for your kind introduction, and good afternoon, dear participants. It's my pleasure to be with you once again as part of this series, which started almost 2 years ago. And before I start, I also would like to thank Sarah for all her help during the preparations for this webinar. So today we are going to talk about laboratory detection of important resistance mechanisms in gram-negative bacteria. Next slide, please. So this is from a 2025 Lancet article. I believe I will show more slides from this article, and I believe it's a very good review of available antimicrobial treatment options against highly resistant gram-negative organisms. So this picture depicts the various resistance mechanisms the gram-negative organisms encompass. And due to many resistance mechanisms, they can exhibit clinical resistance to almost all antimicrobial agents. So even the recent antimicrobial agents are suffering from resistance mechanisms. Organisms are very quick to adapt and then to attack the antimicrobials. Next slide, please. So during my talk, I will mostly focus on resistance mechanisms against beta-lactam antimicrobial agents. So this is an illustration of the activity of spectrum of various beta-lactam antimicrobials. On the left, we have the narrow spectrum agents, beta-lactams or penicillins. And then we have cephalosporins in the middle. And in parentheses, you can see the generation. So, for example, cefadroxil is a first generation cephalosporin, whereas ceftaroline and ceftobiprole are fifth generation cephalosporins. And then we have carbapenems. And beginning of 2000s, carbapenems were considered the last resort antimicrobials for gram-negatives because resistant organisms were so infrequent. But then we started to observe carbapenem-resistant organisms. It started with Klebsiella pneumoniae, then Enterobacter, E. coli, and now carbapenem resistance is frequent among Enterobacterales. Penicillin and beta-lactamase inhibitors. Good examples are amoxicillin, clavulanic acid, and piperacillin tazobactam. We still use them, but they are ineffective against ESBL positive organisms. And this has led to the development of combinations which combine a cephalosporin with an beta-lactamase inhibitor, such as ceftolozane tazobactam, ceftazidime avibactam, and more recently, cefepime and metazobactam. And one of the latest antimicrobials which have been introduced into clinical practice is Astrionam avibactam. So almost each year we are seeing now combinations, but not new agents, combination of old agents to address one particular resistance mechanism. And Astrionam avibactam has been developed to address metallo-beta-lactamases, which I will mention later on. And now we also have carbapenems plus beta-lactamase inhibitors to overcome resistance mechanisms which affect carbapenems. I also added here cefiderocol, even though it is not an combination agent or it is not a new agent. It is it's considered a cephalosporin, a new cephalosporin, but it has a unique way of exerting its activity. Therefore, I put it separately, and I also put it at the last of the table because it shows a very wide spectrum against gram-negative organisms with various resistance or mechanisms. Next slide, please. So most of us are well aware of how an ESBL or positive organism would look like when we test it with phenotypic antimicrobial susceptibility testing. Most ESBL positive organisms are resistant to penicillins and also cephalosporins. So third generation cephalosporins are affected by ESBL enzymes. But carbapenems are saved, they are spared. So most ESBL enzymes are not able to hydrolyze carbapenems. So historically, we were able to treat ESBL positive organisms with carbapenem antimicrobials. So we have observed outbreaks of ESBL positive organisms in 1990s, in early beginning of the 21st century, so in the first decade, but then usage of carbapenems against ESBL positive organisms has led to the emergence of carbapenem resistant organisms. And these were, as depicted on the below bar, NDM, KPC, OXA48, which were resistant to all different classes of beta-lactam agents. So they were, of course, organisms producing carbapenemases were resistant to carbapenems. So to put it into context, next slide, please. I have prepared a series of tables, and you can also check the original article, but these are adapted tables. I try to simplify them to show the various beta-lactamases in Enterobacterales and their clinical relevance. So on this slide, what I indicated with bold font type are the ESBLs. So they are in Ambler classification A and D, but most of them are in Ambler classification A. So here you see most ESBLs are belong to this class, and they include SHV, TAM, and more importantly, CTXM15 type ESBLs. Next one, please. So if we focus on this group, in the same group, there are different beta-lactamases. So the first one could hydrolyze penicillins and early cephalosporins, while the latter can hydrolyze extended spectrum spectrum cephalosporins, which means oxyimmunocephalosporins such as cefotaxime, ceftriaxone, and ceftazidime. So the origin was TAM and SHV group beta-lactamases, but then they were mutated, for example, from TAM1 and TAM2 with mutations. TAM3 arose, and this was in 1985, and this was the first ESBL within TAM group, or from SHV1 with mutation SHV2 emerged, which was again the first ESBL in SHV group. But CTXM was an ESBL from the very beginning. So the first ESBL identified in 1989 was an ESBL. Next one, please. So today, the most prevalent enzyme is CTXM15, and this enzyme is coded on plasmids, meaning it causes outbreaks due to the spread of the plasmid from between organisms. And sequence type 131 E. coli is strongly associated with this plasmid. So we observe ESBL positive organisms in this lineage, ST131, and these usually cause urinary tract infections. And what is worrying with this plasmid is that it doesn't only contain the ESBL enzyme, but it also contains resistance mechanisms against aminoglycosides and fluoroquinolones. So we lose these two important antimicrobials against urinary tract infections when the resistance mechanism is carried on these plasmids. Next one, please. So this is the epidemic strain, which is still in circulation throughout the world, and also more important for us in Europe and UK. So this epidemic clone is carrying resistance mechanisms against aminoglycosides, beta-lactams due to ESBL enzymes, CTXM15, fluoroquinolones, and trimsulfa, and tetracycline. So we lose most of our therapeutic options if the isolate is an E. coli ST131. So this is why it is so important to detect these organisms and try to stop the spread. This is why laboratory detection of these isolates is important. Next one, please. So some information about the epidemiology. Before the year 2000, the most frequently encountered ESBLs in laboratories were TEM and SHV mutants. And these used to cause large hospital outbreaks, and they were largely hospital acquired and mostly observed in Klebsiella species. But then in 2000, the epidemiology started to shift from TEM and SHV to CTXM, and CTXM15 is still the most frequent ESBL type, and it is not only causing hospital-acquired infections, but also community-acquired infections. And it is not only associated with Klebsiella pneumoniae, but also with E. coli. And considering that E. coli and Klebsiella pneumoniae are two organisms which every healthy human being is carrying in the intestine. So it is very important to stop the spread again, because otherwise healthy individuals become colonized with these ESBL positive organisms, and the spread of these organisms continues even in the community. Next one, please. So it took not even 10 years to change the color on these resistance maps. So this is ESBLs in Europe. So in 2005, most of the countries were green, or at least yellow to orange, meaning the resistance percentages were below 25. But if you look at the bottom for E. coli, all those green colors disappeared and replaced with either yellow or orange or even with red, meaning resistance rates reached levels between 25 to 50%. And for Klebsiella, we see dark red, burgundy, meaning resistance beyond 50%. So within a few years, the epidemiology in Europe has changed, and these ESBL positive organisms took over. And this is why we are now facing carbapenem resistance, because the patients then were treated with carbapenems when we realized that we lost third generation cephalosporins. Next one, please. So some information about laboratory detection of ESBLs. Actually, I will not say anything new, but I will repeat the current evidence which works well for detection of ESBLs. And it is never too late, even if the frequency of ESBL positive organisms in your laboratories is high, it is never too late. You should detect these and inform infection prevention and control department in your hospitals to work together. So we should never give up on detecting these organisms. So UCAST has published a very important document, but it was in 2017, and since then it has not been renewed. That's the problem with this document. Some parts of it have been outdated, I should admit, but some parts of it are still valid even for today. So the process to renew it is ongoing, and the aim is to have a new version early next year. So keep an eye on this if you are interested. So UCAST will update this document, and you see the reference at the bottom of the slide. So in this document, various resistance mechanisms are mentioned, and at the beginning of each resistance mechanism, there is this table. Why is it important to detect this resistance mechanism? Is it required for clinical antimicrobial susceptibility categorization? It means if we detect, for example, in this case, ESBL, are we going to change our susceptibility category from S to R, from susceptible to resistant, or from I to R? According to this table, no. It doesn't change our susceptibility category. We are going to report whatever we measure. So if it is S, we are going to report S. If it is I, we are going to report I, because the renewed UCAST clinical breakpoints are taken into consideration also these resistance mechanisms, meaning the clinical breakpoints are sensitive to detect the effect of these enzymes, and the clinical breakpoints are able to separate treatable from non-treatable infections. So we do not have to rely on the resistance mechanism itself, but on the susceptibility category. But the detection of resistance mechanism is important for two aspects. One is infection control purposes. So within the hospital, within the healthcare setting, it's an alarm to detect such organism, and also for public health purposes. We should not let this resistance mechanism spread in the community. Next one, please. So some theoretical information about ESBLs, because we need this information to shape our laboratory strategy. ESBLs hydrolyze second and third generation cephalosporins, and also fourth generation cephalosporins, such as cefepime and cefpiron, but not cefamycins. So good examples in this group are cefoxtine and cefotetan. So if you observe and isolate resistant to ceftazidime and cefepime, but susceptible to cefoxtine, so this is very likely an ESBL. Next one, please. So they have great spread. They have great potential to spread because resistance is localized on a transferable plasmid. And this is why it is important to distinguish this from similar resistance mechanisms. For example, I will not mention all, but the first one, de-repressed chromosomal MC beta-lactamases. So this is not plasmid-mediated resistance mechanism. This is chromosomal. This is specific to this very isolate, and it cannot be transferred because this is due to the changes in the chromosome in this isolate. So usually this kind of isolates do not cause widespread outbreaks. Of course, they can spread, but usually the outbreaks are limited. But if the resistance mechanism is plasmid mediated, then the problem is bigger. So we should be able to separate these various resistance mechanisms. Next one, please. So the strategy should be to screen suspicious isolates or to screen for possible ESBL positive organisms and then to confirm them. So most frequently observed ESBLs, TAM and ASHV. So these usually show obvious resistance to ceftazidime, whereas usually variable resistance to cefotaxime and ceftriaxone. But CTXM, so CTX comes from cefotaximase. So we always expect cefotaxime to be resistant because it's an cefotaximase. So they show obvious resistance to cefotaxime and ceftriaxone, but they can be susceptible to ceftazidime. And this shows us that ESBLs may show different phenotypes. It may be susceptible to cefotaxime, but resistant to ceftazidime or vice versa. And this also changes our algorithm to detect them. All ESBL types show resistance to cefpodoxime. So using cefpodoxime provides us with very high sensitivity, but there are many other resistance mechanisms which also result in cefpodoxime resistance, meaning if we rely only on cefpodoxime, our specificity will be very low. So we should combine all these. And then if we still think it is suspected of being an ESBL producing organism, we should proceed to confirmation step, in which we usually use a cephalosporin agent, and we want to see the synergy with clavulanic acid, because if clavulanic acid can inhibit the effect of ESBL, cephalosporin can regain its activity. So this is the main idea behind confirmation tests. Next one, please. So these are recommendations from Public Health England. It is their SOP for detection of Enterobacteriaceae producing ESBLs. So they recommend to use, especially for hospitalized patients, cefotaxime and ceftazidime on the first line panel, unless cefpodoxime is tested. So if you are testing cefpodoxime, then it is sensitive but not specific. But if you add also cefotaxime and ceftazidime, then you add specificity in your testing. Since there are large differences in MICs, minimum inhibitory concentrations of cefotaxime and ceftazidime, we should combine these agents in our testing panel. Because as I said before, one of the agents can be S, whereas the other is R. We should test both of them to detect resistance. Next one, please. And for community patients, the recommendation is to test for cefpodoxime, because here we do not expect much resistance, and including cefpodoxime might be a good strategy here. Next one, please. So for confirmation, the UK recommendation is to combine cefpodoxime with clavulanic acid. But there are some species such as Enterobacter, Strobacter, Serratia, Morganella, Providencia. These strains, these organisms have chromosomal beta-lactamases, which require that you, instead of cefpodoxime, you test cefpirom, a fourth-generation cephalosporin, because third-generation cephalosporin may produce false positive results if coupled with clavulanic acid. These have inducible AMP-C beta-lactamases, and usually fourth-generation cephalosporin, such as cefpirom, is not affected of MC, and it is therefore more reliable to use cefpirom instead of cefpodoxime. Next one, please. So what do we do and what you can do? Of course, we can use the results of a patient to find suspected isolates of producing ESBL. So the patient's susceptibility test results can serve as the first step for screening. But what if we screen for patients for colonization or for carriage? So in this case, the best sample is rectal swabs. And you can either prepare the medium in-house in your laboratories. So one option might be using McConkey agar and adding cefotaxime 1 milligram per liter. If there's growth on this agar, then the various colony types should be examined further. And the other option is to use commercial agars, such as Chrome, Chrome-ID, Brilliance, Chrome-agar ESBL. And if you are using these, the color of the colony is helpful in identification, and also these limit the growth of other organisms in the patient's flora. So it is highly helpful in eliminating the organisms which we are not interested in. So if you observe growth on these agars, you proceed further. Next one, please. And if you are using patients' AST results, then this is a very useful table. So these are the current UCAST clinical breakpoints for cefpodoxime, cefotaxime, ceftriaxone, and ceftazime. What you see on the left, highlighted in red, is that they all share the susceptibility breakpoint of 1. So if you observe any MIC value which is more than 1, it means it is screen positive, meaning it can be ESBL positive. So look further, investigate further. And if you look to the right, zone diameter screening cutoffs. So if you perform this diffusion, here things get a little bit complicated because susceptibility breakpoints are different than screening cutoffs. So susceptibility is something, screening is something else. You can report susceptibility based on clinical breakpoints as indicated in this table. But if you want to identify suspected organisms, then use the screening cutoff indicated or highlighted in red color. These are more sensitive. And as I said before, these are taken from the document published in 2017. So there may be some discrepancies between clinical breakpoints and screening cutoffs. So I think all of these will be addressed in the next version. Next one, please. So if you identify your suspected organisms, then you can proceed with this algorithm for phenotypic detection. So it starts here. you use the screening criteria to identify the suspected isolates. And then you proceed according to the organism type. In group 1, we have the strains, organisms such as E. coli, Klebsiella, Proteus, Salmonella, and Shigella. None of them have inducible AMP-C. But here we have other Enterobacteriales species such as Enterobacter, Citrobacter, Morganella, Providencia, Serratia, Hafnia. They all have chromosomal AMP-C, and therefore our confirmation algorithm changes. Here we can use ceftazidime and cefotaxime, but here we cannot use them due to chromosomal AMP-C. Here we have to use a fourth generation cephalosporin. Here the recommended agent is cefepime, and UK SOP recommended cefpirom. So I am following the UCAS recommendation, and we in our laboratory use cefepime plus clavulanic acid for this kind of organisms. Next one, please. So I will show some pictures of available tests. I'm sure you are familiar with most of them. So this was the first method developed for detection of ESBLs, double disc synergy test. So amoxicillin clavulanic acid in the middle, and on other, at both sides, we have cephalosporins, third generation cephalosporins, here ceftriaxone and ceftazidime. And you see the synergy, which is due to the clavulanic acid. So ceftriaxone alone is resistant. Amoxicillin has some activity, and clavulanic acid diffusing towards ceftriaxone is causing this enlargement of inhibition zone, also called synergy. So here we have synergy with both agents, ceftriaxone and ceftazidime, also here. And in this example, ceftazidime alone is very likely susceptible. I don't know the inhibition zone diameter, but it looks a very large zone. But here we have cefotaxime, which is almost 6 millimeters due to these colonies here. But together with amoxiclav, it shows this synergy. So it is positive for ESBL. So this is very likely a cefotaximase. So ceftazidime is not affected, but cefotaxime is hydrolyzed. So we can all use this in our laboratories as a confirmatory test. And the recommendation is to place discs 20 millimeter center to center. But this may not be optimal for all organisms. Sometimes you have to optimize it a little bit. But this is the general recommendation. Next slide, please. Here, ceftazidime plus ceftazidime clavulanic acid. Clavulanic acid has no activity. Both have 6 millimeters. We cannot call this an ESBL. And here, cefotaxime and cefotaxime clavulanic acid, again, the difference is less than 5 millimeters. So this, we cannot call this ESBL. So in this case, further tests should be done, because maybe we have ESBL, but if coupled with another resistance mechanism, such as AMP-C, then we are not able to detect it. So in this case, I would go further with different laboratory methods to see if it has AMP-C, and I will show later. Next one, please. Here with ceftazidime, the difference is less than 5 millimeters, but with cefotaxime, it's more than 5 millimeters. Again, emphasizing why we should use both of the agents. Next one, please. We can also use gradient strip tests for detection of resistance mechanisms. Here we have ceftazidime plus ceftazidime clavulanic acid. Ceftazidime has an MIC alone of 12 milligram per liter, but 12 is not an dilution step. We should round it up to 16, and 16 versus 0.25. So the definition is to observe more than or equal to 8-fold dilution, or in other words, 3 dilution steps reduction in the MIC. So ceftazidime alone has an MIC of 16, and it is reduced to 0.25 with the addition of clavulanic acid. And this corresponds to 64-fold reduction, or we can also call it 6 dilution steps reduction. So this is ESBL positive. Through the years, I realized that this 8-fold reduction is not clear to everyone. This is therefore I put some examples here. It's not difficult, but it's best to see it once at least. Next one, please. Here another example, cefotaxime, MIC of 1. So the ellipse is thought to be cutting the strip at around 1. And then cefotaxime clavulanic acid 0.094 rounded up to 0.125. This is the doubling dilution, because doubling dilutions are counted up and down from 1. So 1, 0.5, 0.25, and then 0.125. These are intermediary concentrations, which we do not use in broad microdilution in reference testing. Therefore, also with gradient strip tests, this should be rounded up to the doubling dilution. So here what happens is we also see synergy due to clavulanic acid, and this is called phantom zone. So if you observe it, it is indicative of ESBL production or indicative of synergy. But if we look here, how much reduction we obtain, it is also 8-fold or 3 dilution steps. So even without the phantom zone, it is ESBL positive. Next one, please. Another example on why we should use both agents. So sometimes one of the agents is totally susceptible. So here in this example, the bottom strip, ceftazidime. Ceftazidime MIC is less than 0.5. So it is actually screen negative. I remind you that screening cutoff was 1 milligram per liter. So less than 0.5 is too low. It would not classify as suspected, but with the addition of cefotaxime, we are able to detect the ESBL production. Next one, please. 4 more examples, just to emphasize that if you see this synergy, so again, phantom zone, or also here, even though cefotaxime is more than 16, we see this synergy. So these are examples on ESBL production. And if the MIC is more than on both sides of the strip, then we do not call this negative, but we call this indeterminate or non-determinable. And we should do more tests. Next one, please. So these are pictures from our laboratory. We do not perform broad microdilution for ESBL, but this was a project, and for this project we tested these panels. So here you have cefotaxime. Cefotaxime is more than 64, and cefotaxime plus clavulanic acid is less than 0.06. So it's a very clear ESBL producing organism. Ceftazidime alone 32, ceftazidime clavulanic acid 0.5. So this is another method which can be used, but this is not practical. and this is highly costly. So I wouldn't recommend it routinely, but just showing you because it's possible. Next one, please. And another example, here we have with cefotaxime, we are able to detect ESBL, and with ceftazidime, we cannot ascertain it's an ESBL production because MIC is 4 versus 1. So it's 4-fold reduction or 2 dilution steps. So with this, it is negative, but with cefotaxime, we detect it. Next one, please. So I put most of my energy into ESBLs because I believe the whole philosophy can be explained with ESBLs. And then I will also talk on MC and carbopenemases, and the principles will be the same. So it will be now easy to cover these two resistance mechanisms. Starting with MC, we see that it's the same. You do not use it for susceptibility categorization, but acquired MC is important to detect because acquired means it can also be spread. It can also be transferred to other organisms. Next one, please. So MC enzymes are located in Ampler classification group C, and MC enzymes hydrolyze cephalosporins. Next one, please. So they hydrolyze cephalosporins, including third-generation cephalosporins. So now it makes sense why we do not use third-generation cephalosporins for organisms with chromosomal MC, because third-generation cephalosporins are hydrolyzed by these enzymes. But fourth-generation cephalosporins are able to withstand MC enzymes. They are poorly inhibited by classical ESBL inhibitors, such as clavulanic acid, but they are inhibited by other substances, and we are going to use them for diagnostic purposes. E. coli and Shigella have chromosomal MC, but it is not of clinical concern because it is only produced at trace levels. Whereas Enterobacter, Citrobacter, Morganella, Pseudomonas, Providencia, and Serratia, they also have chromosomal MC, which is inducible. So if the patient uses, for example, third-generation cephalosporins, then this enzyme is induced very quickly. And even if the patient has a susceptible isolate on day 1, after 1 or 2 days of treatment with a third-generation cephalosporin, this enzyme may be induced, and then we can isolate a resistant organism from the same patient. And this is why it is important to test these isolates again and again from the same patient if we observe it, if we isolate the same organism from the patient. Next one, please. For screening, we can use cefoxtin, and MIC of more than 8 is indicative of MC beta-lactamase, and we can also use cefoxtin disc. In this case, corresponding zone diameter is 19 millimeter. MC is inhibited by cloxacillin and boronic acid, and we use this for diagnostic purposes. So next one, please. So here we have the organisms which lack chromosomal MC. So E. coli, Klebsiella, Proteus, Salmonella, and Shigella. So these are the group 1 organisms in the ESBL algorithm. So we can look for synergy with cloxacillin. So the same philosophy For ESBL, we looked for synergy with clavulanic acid. And here we look for synergy with cloxacillin because it is inhibiting MC. Next one, please. So there are various commercial tests. This is one of them. Here there are 3 tests, and these include cefpodoxime plus an MC inducer. Another disc contains, in addition, ESBL inhibitor, and the third one, the C disc, contains MC inhibitor. So when MC is inhibited, the agent cefpodoxime regains activity, and this is confirmatory for MC beta-lactamase. So if with the addition of inhibitor, the enzyme is inhibited, then the responsible resistance mechanism is MC. Next one, please. And MC can also be detected by gradient strip tests. Here, the inhibitor is again cloxacillin. On one side, we have cefotetan. On the other, we have cefotetan plus cloxacillin. So cloxacillin inhibits the enzyme and activity is regained. This is typical for MC beta-lactamase. Next one, please. So if we proceed with carbapenemase production, the importance is the same. Next one, please. And carbapenemases are found in 3 different groups. Important carbapenemases can be found in 3 different groups. Group D include OXA48 oxacillinases. Group A has KPC, and in group B we have metallo-beta-lactamases. More importantly, IMP, VIM, and NDM. Next one, please. So these were emerged not very far from today. IMP was the first one, 1991. Then came VIM, 1997. And the same year, KPC was detected in USA, 1997. Then we found OXA48 in Turkey in 2001. And in this group, the last one was NDM, New Delhi metallo-beta-lactamase, detected in 2010. And now they are widespread throughout the world. Next one, please. So I will show a few slides about how things have changed very quickly. So this is from 2001, describing the KPC enzyme. Next one, please. So in 2001, it was only in New York, 1 state only. And after 10 years, it was all around USA. Next one, please. This is the first paper describing NDM. And when this paper came out, next one, please, it was very certain that the detection of metallo-beta-lactamases would change everything, because these were responsible for high-level resistance to all beta-lactamases. So with metallo-beta-lactamases and with KPC, we lost all our beta-lactamase antibiotics. So it was called the quiet before the storm. But this is 2005. And next one, please. And this is 2012. Only after 7 years, the same authors called it, here is the storm, carbapenem resistance in Enterobacteriaceae, because in 2012 already metallo-beta-lactamases were all around the world. Next one, please. And why this was called the storm? Because these isolates were carrying plasmids, conferring resistance to almost all antimicrobials. So I don't like the term pan-resistant, but most of these isolates were pan-resistant with considering the capacity the laboratories had. So all tested antimicrobials were resistant. Next one, please. And analysis of one such isolate revealed the resistome. Next one, please. So this is the list of various resistance genes in an NDM1 producing E. coli. So these isolates are now in all our hospitals and also in the community. So treatment of these infections have become such a challenge, I should say. So what can we do in our laboratories to detect these? Next one, please. Next slide, please. Thank you. So again, there are clinical breakpoints for clinical categorization as S and R, but there are also screening cutoffs which are helpful for us to detect carbapenemase producing organisms. As you see, they are quite lower than the susceptibility breakpoint. So for meropenem, an isolate with an MIC of 2 is susceptible, but it may be an carbapenemase producing organism. So it is therefore very important to use the screening cutoffs and not the clinical breakpoints for resistance mechanisms. This is for treatment, and the screening cutoffs are for resistance mechanisms. And on the right, we also see the screening cutoffs for this diffusion. And here for meropenem, I have put 25, but there are two options, either 25 or 28. To be more sensitive, I would recommend to use 28, and I will show it in the next one. Next slide, please. So here, starting with a meropenem screening cutoff of 28, we can detect almost all carbapenemases in Enterobacterales. And this table is just a recommendation for identifying different enzyme types. For example, KPC is showing synergy with boronic acid, MC with boronic acid and cloxacillin. Metallo-beta-lactamases show synergy with dipicolinic acid, and we can use these in our diagnostic tests. Next one, please. Examples on, the title is wrong here. It is not gradient test, but this is combination disc test. Sorry for that. We have commercial combination discs. So on the right, it is easily readable. So we have meropenem disc only, and then meropenem disc combined with different inhibitor substances, such as boronic acid, dipicolinic acid, and cloxacillin. So in this case, meropenem doesn't show any synergy with any of these substances, but resistant to temocillin. So this is highly indicative of OXA48, because OXA48 enzyme doesn't have any specific inhibitor. So if you do not serve any synergy or inhibition with any of these substances and you see temocillin resistance, this is very likely OXA48. Next one, please. So in the UCAS document, there's an additional table helpful for these interpretations. So looking at this table, you can suspect with a very high level of correctness what enzyme it might be. For example, dipicolinic acid is So metallo-beta-lactamases show synergy with typicolinic acid or EDTA, but not with anything else. Next one, please. So this is how this information may be used with gradient tests. So here we have imipenem plus EDTA. Imipenem alone is more than 256, but with the addition of EDTA, it is less than 1. This is typical metallo-beta-lactamase. The enzyme is inhibited with EDTA. Next one. Here we have ertapenem and ertapenem boronic acid. The picture and the text are not corresponding to each other. So I think I put the wrong picture here again, sorry. But we can use boronic acid for KPC. But the thing here is boronic acid is also an inhibitor of MC beta-lactamase. So it is not enough to call it an KPC, even if we see inhibition with boronic acid. Next one, please. There are some commercial tests which can be used for carbapenemase detection. For example, this one here is carba-NP test. It can be used, it can be prepared in-house. So these are pictures from my laboratory. All the regions can be obtained and the test can be set up in-house. But you can also buy it from commercial vendors and you can use a kit instead. Next one. And this is a test called SIM, or now modified SIM, MSIM, carbapenem inactivation method. So this only requires carbapenem disc and the organism. So it is very cheap and it is highly reliable, especially the modified version. And it is now included in the CLSI M100 document. So this is a well described and well validated method. So this can also be an option because it doesn't cost anything. The disadvantage is that it is slow. It requires an overnight incubation, but it is reliable. And here we see that if the organism has carbapenemase, it inactivates the meropenem component in the meropenem disc, and then we see that meropenem disc loses activity. More information is in CLSI M100 document or in the original publication. Next one, please. And of course, now we also have immunochromatographic tests. So this is one of them, but there are other products in the market. This is Coris from Belgium. There's also NG from France, which I see very frequently when I visit laboratories. So these kits can detect those 5 important carbapenemase types, IMP, VIM, NDM, KPC, OXA48 in 15 minutes. The thing is, they are a little bit costly. So it is not for everyone. It is not for every isolate, but this is a very rapid method. So if the patient requires quick results, this might be an option. Next one, please. I will finish with colistin resistance. Here, the only difference is that it is required for susceptibility categorization, because here colistin resistance means colistin susceptibility. So in this case, if we detect resistance, of course, we have to report it as resistant. So, and it is also important for infection control purposes and public health purposes. Next one, please. One important thing here to consider is that some Enterobacteriales species are intrinsically resistant to colistin. For example, Proteus, Morganella, Providencia, and Serratia. So the things I'm going to mention do not apply to these because these are intrinsically resistant. And here, the resistance mechanism is not acquired, or it is not due to mutations. It is chromosomal. Next one, please. So we are interested in acquired resistance mechanisms. So these are either with mutations or with acquisition of plasmids. Next one, please. So here we see the outer membrane, and colistin requires negatively charged lipid A to be able to exert its activity. And if there are mutations which result in the loss of negative charge in the lipid A in the outer membrane, then colistin cannot act and the organism becomes resistant to colistin. Next one, please. So this can happen in 3 ways, in 3 different mechanisms are responsible. So chromosomal mutations, regulatory system, and also plasmid-mediated resistance genes. And among these, MCR genes are more the most common ones, most widely known resistance mechanism. Next one. So I will go quick here. These are reports of MCR1 producing isolates in humans, animals, and both humans and animals. So MCR1 was spread in Europe. Next one, please. And colistin resistance was mostly observed in carbapenem-resistant organisms. So on the right, you also see the names of various enzymes. And this is a paper from 2017. So in the last almost 10 years, colistin resistance has increased in carbapenem-resistant organisms. And next one, please. And these MCR1 to MCR9, but now we have more, have spread all around the world because this is spread on plasmids. So we should be able to detect these isolates also in our laboratories. But we should not focus only on if it is MCR or if it is chromosomal mutation. We should be able to detect if there is resistance or not. So this is the current UCAST recommendation. Next one, please. So UCAST has some recommendations on colistin susceptibility. So in short, it is regular standard broad microdilution. I will not go into detail because many options were tested, and now the recommendation is to perform broad microdilution. Next one, please. So these are the clinical breakpoints as of today with UCAST and CLSI. So the 2 committees are almost in accordance now. The difference is UCAST only recommends broad microdilution, whereas CLSI has some additional methods. And this is called colistin broad disk elution and colistin agar test. And I will shortly show them. Next one, please. So only if you cannot perform broad microdilution, these 2 methods can be considered, but UCAST recommendations is broad microdilution. So here, the principle is very simple. You add colistin discs into broth. So colistin sulfate discs, 10 microgram. 1 disc in tube 1, 2 discs in tube 2, 4 discs in tube 4. and this is the growth control. Then you add the organism, and then you incubate and look for growth. And these discs elute from the disc and correspond to 1, 2, and 4 milligram per liter in the broth. So it is a very rough way of performing broth microdilution, but it is a validated method and recommended by CLSI. Next one, please. And this is the cholesterol agar test. So here we have various concentrations of cholesterol in the agar. So on the left, we have no cholesterol growth control, then 1, 2, and 4 milligram per liter. This is the same as, almost the same as agar dilution, but instead of preparing and pipetting a certain amount of liquid, here streaks are made. So this is a deviation from the standard. And again, more details are available in CLSI document. Next one, please. So if you cannot perform broth microdilution in-house, you can purchase commercial products. For example, these two, SenseTiter and Micronote, have cholesterol among other antimicrobial agents in their multiple agent panels. But next one, please. You can also use one of these single agents. They are available from UMIC and COMASP. So these kits only contain cholesterol, for example. So this is a kit for cholesterol MIC determination. So if you are only interested in cholesterol, you can also consider using these products. Next one, please. So since broth microdilution is the only valid method for cholesterol susceptibility, I will speak also about skipped wells and then finish my presentation. So when we perform broth microdilution, we sometimes see skipped wells. And for this, there are some rules. For example, here, one skipped well. So we can either read the MIC here as the highest MIC value, or we can retest the isolate. But if we observe more than one skipped wells, here 2 and here 3, then we should call this test as invalid. I will show some examples from our laboratory. Next one, please. Here, an E. coli, two wells were skipped. So should we read this here, or, sorry, at 0.5? Can you please click, Sarah, as I speak? Yes, 0.5 or 1 again, or at 4 again. No, please click again, because this is invalid, and we should retest if results of cholesterol susceptibility is required. Next slide, please. You can click 3 times. So again, this time one single well, is it 4 or 16? If I cannot repeat the test, I should report as 16. But if it is important for the patient, then I should repeat. Next one, please. So the isolate has been retested. So actually, the MIC was 16. Next one, please. And you can click 5 times. So 3 skipped wells. Should I read 4 or retest or invalid? Of course, invalid. Next one. And since the first test was invalid, I retested it, and this time I got 0.25. So this actually shows reporting the highest MIC is not always the best option, because you are losing the treatment with cholesterol. So if you see invalid results, best is to retest the strain. Next one, please. This was a very difficult isolate. You see that the growth is showing a trailing effect. Even here at 4, I see some haziness. So the first well showing no growth at all is 8. But is it right to read it at 8, or should I go at 4 or even at 2? So we retested the isolate. Next one, please. And we observed the very same MIC, even though we tested on another panel type. So different MIC values, because these are 2 different panels. But on both of them, we observed the same phenotype, and we tested this as 8, and it was resistant. The patient couldn't use colistin. Next one, please. So as a summary, now we have many therapeutic options effective against various resistance mechanisms. For example, avibactam, and in many countries, ceftazidime avibactam is available. So it is very good for KPC, ESBLs, but it is ineffective against metallo-beta-lactamases. So right now, or until very recently, there were no agents that were effective against metallo-beta-lactamases. This is from 2020. Next one, please. And this is from 2022. Cefiderocol has entered the market in some countries, and it is effective against many resistance mechanisms. And then we have colistin. Unless resistance is observed, it can be an option. And this is from 2022. One more, please. And this is from the Lancet review, which I showed as my first slide from 2025. This is the most recent one. And now you see we have new agents here. Aztreonam avibactam, which shows effectivity also against metallo-beta-lactamases. And here we have cefepime and metazobactam. Here we have sulbactam, durlobactam. So we have new agents, but what is important for the laboratories is to detect the susceptibility correct, and also to detect the resistance mechanism. Because if we know the resistance mechanism, we have some options for cure. So this is why it is important to detect these resistance mechanisms in gram-negatives. Thank you very much for your attention.
Дуже дякую за цікаву та змістовну доповідь.
Thank you very much for very interesting and meaningful lecture. Really very intensive and interesting material for our colleagues from bacteriological labs. And since we exceeded our time, let's maybe shorten the Q&A session. And first of all, I would like to mention that this webinar, like the others, is recorded and it will be uploaded to the YouTube channel of the UPHC. So please subscribe to our resources, rewatch the webinars. They will be on the special playlist. And many participants actually saved this playlist on their desktops. So I'm urging you to do so. And with regard to the questions, I saw in the chat one good question. Maybe Dr. Karatuna, you can tell us. Do you know when the guideline on phenotypic determination of carbapenemase in Pseudomonas and Acinetobacter will be updated? Do you know when?
For Acinetobacter, I don't think much will change, because the resistance mechanism is mostly due to non-enzymatic mechanisms. So the responsible mechanism is not carbapenemases. It is more important for Pseudomonas aeruginosa. There we can have IMP or VIM producing isolates. And I believe the changes will be available early next year. So this is earliest, because I know that the work is ongoing. And whenever UCAST publishes something, they first put it on for consultation. So you will receive information about that a consultation is available before the final publication is published. So, and since we do not have the consultation yet, I don't think it will be earlier than next year.
Дуже дякую за відповідь на питання.
Thank you very much for your
Колеги
answer.
запитують наступне.
And the
Де
colleagues
мають
are
визначатися
asking
гени резистентності? На
where
національному рівні, національному
resistance
лабораторії
genes
або в лікарні?
should be identified at the national level in the UPHC or locally in a hospital?
Of course, it depends on the capacity. If you have it, you can do it. But what I can share with you, our practice here in Sweden, if an organism is resistant to carbapenems, we send it to Public Health Agency of Sweden for genotypic testing. So we have the capacity, but we don't perform it. Instead, we send it to the reference center for genotypic testing, which is Public Health Agency of Sweden. And they collect all these isolates from all over the country, and they perform tests, which we cannot do because they also perform whole genome sequencing, and they also look for relationship between different strains. So what we can only do could be to detect the resistance gene, for example, to call it NDM. But since they do it in a more sophisticated way, they also look for relationship between strains collected from different cities, from different hospitals, and it provides more important data. They have a better idea of circulating isolates. For the patient, I think it is enough to perform reliable susceptibility testing, because this determines if the patient receives carbapenems or not. And if it is carbapenem resistant, then it is due to carbapenemase production in most of the cases. And so it should be considered as carbapenemase production for infection prevention and control. And genetic data can come later on from the public health institution.
Дуже дякую за таку практичну відповідь.
Thank you very much for very practical answer. And the last question for today, and we will finish. I'm not a bacteriologist. Maybe I will interpret not right it. But the question is, how long does it take for gram-negative organisms to retain resistance, maybe in certain populations or in certain areas?
If there are patients nearby with plasmids which are transferable, so it is within days that nearby patients can get colonized and then can get infected with these organisms. So it is it depends on many factors. Therefore, I cannot give any clear answer. But if the infection prevention measures are not followed strictly, then of course the organism will spread to other patients, and then we are going to observe an outbreak in the healthcare facility. So I would say within days, we would start to see new patients.
Дуже дякую за вашу відповідь. І ми будемо
Thank you very much for your answer. And we will conclude our session. I would like, on my behalf and on behalf of the participants, to say words of gratitude. And actually, people in the chat write their words of gratitude, saying that the lecture was very interesting. We had more than 300 people at some point. So everybody is grateful, and I would like also to join them and remind you, dear participants, that the video recording will be available on the YouTube channel of the Ukrainian Public Health Center. In order not to miss the future announcements of such webinars, please subscribe to the Facebook page of the UPHC, and the announcements will be there. Thank you very much, Dr. Karatuna. Thank you, colleagues from CDC and ICAP, and thank you, translators. So I wish everybody to have a nice day and peaceful sky.